Good morning, everyone, and thank you, the HFSP organizers, for giving me this opportunity to really talk about my work. I'm from the muscle group at Yale University, where we primarily study early animal cell type evolution, and my project primarily focuses on the question of how the first animal cell types talk to each other. By that, I mean different organisms, a life form on Earth, have to interface with the environment. But what's unique about animal is that they also have different cell types in their body that also have to talk to each other using transmitters. And when we think about how a cell that's just crawling on a surface versus how a worm crawls, the fundamental difference illustrates the principle of division of labor that took place at the dawn of animal multicellularity. By that, I mean at the molecular level, a single-cell eukaryote has the potential to have all the correct machinery to detect environmental nutrients and then mobilizing its cytoskeleton machinery to crawl towards it. While when we think about animal, we usually associate the role of sensation to specialized cell type like the neuron, which then integrate these signals and then transmit information using neurotransmitters to various effector cells, one of which are muscle cells that are organized in very regular structure along the body to perform large-scale contractile motions to generate locomotion. Now, neurons and muscles are incredibly complex cell types, and this is definitely not how the first animal ancestor organized their physiology and coordinated their behavior. So in our lab, we work with one of our most distant elemental relatives, which are sponges. Sponges are these fascinating aquatic organisms that have only a handful of cell types. They don't have traditional neurons and muscles, but their cell types must also talk to each other to coordinate their physiology. So by really comparing what is shared between sponges and us, we can really then infer how the first animal cell types started talking to each other when animal multicellularity began. So the sponge we study in the lab is the freshwater sponge, Spongella lacustris. These animals are great because they form these packets of stem cell cysts in the winter that we can go out in lakes to collect. And on the bottom, this is what we usually collect from lakes. We can scrape off these cyst-filled stem cells. And we want to work with them. We pop these cysts into water. And after a few days, this white tissue of the whole sponge body emerges. Now, the body of the sponge is optimized for filter feeding and water flowing. Water enters their in-current canal, labeled in blue, and then it passes through this digestive tissue in yellow, where nutrients get absorbed in these ciliated digestive chambers. And then whatever is remaining, like waste, is then expelled through the excurrent canal in red and out through this chimney-like structure. Now, to better study these animals' behavior, we built an optical coherence microscope that allows label-free volumetric imaging of the sponge. Here we have a time series showing that at the minute scale, there are different kinds of behavior mode that these sponges undergo. So you can see that there are some minor twitches in their different canal system, but they also undergo this large-scale deflation response where they completely close up their incurrent and expand their excurrent canal. Now, previous work in the lab have shown that nitric oxide, which you may know is a similar vasodilator in our own vasculature, actually is responsible for this expansion of the excurrent canal system. And what we see is that when we add nitric oxide, there is this drastic expansion of the excurrent system. But nitric oxide, as we all know, is a diffusible gas, and it's not a reliable transmitter that can be stored for release. So I first started this project by testing a bunch of different chemical transmitters in our own body to see if they might act upstream from nitric oxide, or maybe they're responsible for the recovery response. Well, it turns out some of the well-known synaptic transmitters, like glutamate and GABA, and also some of the neuromodulators, like dopamine, serotonin, they don't really elicit much drastic behavioral response in these sponges. Interestingly, we found that some of these non-canonical monoamines, like tryptamine, phenethylamine, and tyramine, which are simple derivatives of amino acids, they're the ones that are able to elicit drastic behavioral response at low micromolar concentration. So what do these responses look like? Well, in the case of tryptamine, I'll first walk through the behavior. So we can see that when we add tryptamine to the sponges, there's this initial constriction of the sponge tissue as quantified by the top right graph. And this initial constriction is then followed by this expansion of the excurrent canal, similar to the nitric oxide response. So we think it might act upstream of nitric oxide. So here we have again our OCM image of the sponge. And we can see that when we add tryptamine, there's this initial constriction of the tissue and then the follow expansion of the excurrent canal. Now, unlike tryptamine, we also saw that for phenethylamine, it actually does the reverse, where it tries to pop up the sponge tissue, actually elevates the sponge, and we think this is the recovery mechanism after nitric oxide. And lastly, tyramine, which is also interesting in the sense that it doesn't induce any response by itself, but actually modulate the activity level of the sponge tissue so that the sponge appears to be more active. And this is very interesting because some of you might know that invertebrate tyramine is analog of our adrenaline in our own nervous system. After this, we tested these molecules against different sponge species that we either could collect in the field or had in our lab, and we showed that this behavioral responses is actually conserved across demo sponges. And for evolutionary distance comparison, these species are related the same way that we're related to fish. So it's about 400 million years of evolution conserved behavioral response. Next, we wanted to really find out how sponge cells are making these transmitters, and they have specialized secretory cells. Because in our own neurons, they have specialized enzymes that metabolize amino acids to make these different monoamines like dopamine and serotonin, and then these amines are stored in vesicles by the action of some transporter. Well, it turns out sponges have a much easier job because the monoamines they use only require a single decarboxylation modification, unlike some of the monoamines we use in our body. But even if they only need a decarboxylase and transporter, previous phylogenetic efforts have not been able to find the canonical decarboxylase or transporter in the sponge genome. So we had to go and search harder. So here is a phylogenetic tree of the large decarboxylase protein family, and we can see that the canonical decarboxylase found in animal neurons is not really present in sponge. But we did find two other previously uncharacterized decarboxylases that are in the broader protein family that are found in other animals, but also these are the ones that are present in sponge but uncharacterized. Similar with the transporters. Now the transporter protein family is this gigantic protein family that goes all the way back to bacteria. And we also found that there were several uncharacterized, potentially vesically localized transporters that's in the sponge genome. So to really test the function of these genes, we teamed up with our colleague using the roundworm nematode C. elegans, where they have a very beautiful behavioral assay where you can actually touch the worm on the head. And when you touch the worm on the head, it undergoes this reversal escape behavior. And this is tyramine dependent. So when you have tyramine, the worm is able to reverse. If you mutate out the worm decarboxylase or transporter, you see that there's this decrease in the escape response, which then can be rescued if you put back in the warm decarboxylase and transporters. We were very happy to see that we put in the uncharacterized sponge decarboxylase. This was also able to partially rescue the behavior of these worms, suggesting that these are indeed functional decarboxylases. And out of the 10 transporters we found in sponge, 2 of them also were able to rescue this behavior. Now, if you remember, I previously mentioned that some of the decarboxylase is found in other animals. Well, it turns out this enzyme, PDX-DC1, which is found also in mammals, especially in the gut and some regions of the brain, is also a novel decarboxylase that has not really been appreciated before in even mammalian physiology. After doing these functional characterizations, we went in and checked, and to ensure that these genes are actually co-expressed in 2 distinct cellular populations of the sponge. Here I'm showing the in situ hybridization where we stain the RNA molecules of these genes, and we see that one set of the transporter and decarboxylase is co-expressed in metabolocytes. These are cells you can think a fusion between hepatocytes and glial cells. They are regulating the metabolic state of the sponge and maybe signaling to talk about the satiety status of the organism. While the other cell type, the nervous cell, has a lot of neuronal machinery for secretion, but they also have an amoeboid-like phenotype where they actually crawl around into these digestive chambers and maybe monitoring the water quality. Okay, so with the secretion machinery found, we then looked at the distribution of these secretory cells, and we can find they're regionalized across distinct areas of the sponge body, where we think this might be the first almost circuit of where you place different volumetric signaling centers in your own body. So the first initiation of paracrine signaling that resembled the ancestral animal communication network. Okay, so now let's move on to the effector state. So how are these sponge cells responding to these monoamines, and what pathway do they activate? Because we have kind of two competing areas here, in the sense that unicellular eukaryotes, they're mostly using contraction for chemotaxis and migration, whereas muscle cells in our body have very distinct organization with dense bodies and also sarcomeres to mediate fast and large force-generated contractions. Well, to turn a long story short, we performed quantitative phosphoproteomics on these sponges treated with tryptamine, where we were able to detect differentially phosphorylated proteins to see what pathway are activated. So after a lot of work and like looking at the pathway, this is a summary of the players that we basically found. So it turns out that sponges are using a mix of unicellular contraction machinery, where they actually use Rho GTPase signaling, classically studied in the context of cell migration. But in these sponge cells, they have tethered that machinery to a lot of the cell adhesion machinery that has been invented at the dawn of multicellularity. So it's almost like a less effective musculature contraction system. Now, of course, we did some drug perturbations, which I will not show today, to indeed show that these, by perturbing these different pathways, you can lead to different behavioral changes upon the treatment of tryptamine. And I'll just show that we also saw GPCRs that are differential phosphorylated in our phosphoproteomics screen, which was very good because it shows that this ability to activate GPCR, which are also the case for monoamines in our body, is conserved in sponges. We then looked at where these differentially phosphorylated GPCRs are expressed. We saw that they are mostly found in the canal lining cells of the sponge, and some even in the secretory cells that are making these monoamines, suggesting maybe some autofeedback inhibition. So when we then looked at the distribution of these different cell types with a whole body imaging of the sponge, and we saw that there's this very curious differential distribution where we have these incurrent epithelial cells forming on the tent, but also covering the body of the sponge, these malpacted cell magenta cells forming an elaborate mesocilial network, and also these excurrent epithelial cells that are lining the excurrent larger canals. We then looked at vinculin staining along the tent as a marker of how cell adhesion is being modified, and we see that with the addition of tryptamine, there's a lot of vinculin foci that appears, and sometimes they look bigger. So this is suggesting that they are modifying cell adhesion, and they're also increasing the phosphorylation level of myosin light chain, which is a marker for contractility. So in summary, we found that sponges have this form of monoaminergic signaling system in their body. They have regionalized secretory cells that might be sensing different environmental cues. These amines are then secreted to effector cell population and mostly modulating the tonality of the canal and performing an early form of contraction. So I began the talk by talking about how the first ancestral animal cell types start talking to each other. And I think from our work in sponges, we can infer that at the beginning of animal multicellularity, there was this ancestral secretory cells that have repurposed nutrients like amino acids, modified them slightly, turned them into metabolites that can act as a organismal specific and intraorganismal but intercellular cues that they can send towards effector cells. And in this case, in our musculature, they kind of took the ancestral constriction toolkit for chemotaxis, they anchored that with cell-cell adhesion machinery, and that's how they can form a large monolayer of tissue and how that tissue can act in a coordinated manner. So at the end, we think now we have a better mapping of how the evolutionary stepwise emergence of multicellular coordination emerged. We changed from a autonomous cell-sensing crawling cell into a animal with different cells sensing the environment and communicating to a larger, more specialized tissue to perform coordinated behavior. And these were then elaborated later on in us, where we have much more complex cell types and much more sophisticated signaling mechanism in our neurons and muscles. And with that, I would like to thank everyone in my lab for supporting my work, and also HFSP for funding this amazing research and letting me have the chance to share with you all. Thank you.